Welcome to the Discover Alaska Lecture Series, brought to you by UAF Summer Sessions and Lifelong Learning. My name is Althea St. Martin, and I'm your host for this summer series. I'm excited to introduce tonight's speaker, Ed Murphy, who will be presenting Going to Extremes, Diversity in Birds. Ed Murphy is a UAF Professor Emeritus and has taught courses in ornithology and animal behavior, data analysis, and natural history during his 30 years at UAF, and for Ollie the last several years. His research has focused on cliff-nesting seabirds in northwestern Alaska and the Bering Sea, and on woodpeckers in the interior. Thank you, Ed, for sharing your bird expertise with us this evening.
Hi, my name's Ed Murphy. I'll be talking about extremes in bird migration strategies today. Talking about a couple of species that embrace the winters here, and then other species that go considerable distances away from Alaska in the wintertime. I retired from the university several years ago. I had a joint appointment in the Department of Biology and Wildlife in the Institute of Arctic Biology. If there's anything that strikes your interest today, feel free to contact me by email, ecmurphy@alaska.edu. I'd first like to just talk about some of my initial discoveries when I first came to Alaska. My early personal experiences started in August 1971 when I arrived in Fairbanks with Gretchen, my wife, and frequent field companion. In the summer of 1972, I did my master's study on Dall sheep in Denali Park. Ken Whitten was also working on sheep at the same time. Our advisor was Dale Guthrie, and we had some good friends helping us. In the summer of 1973, Gretchen and I went to the Brooks Range on the Alaton and the Killick Rivers, and there we did plant surveys in the proposed Gates of the Arctic National Park, helping Dave and Barbara Murray. In the summer of 1977, after finishing school outside, I came back and we went out to Cape Thompson in northwest Alaska. It's a site with high numbers of cliff nesting birds, approximately 400,000 murres and 30,000 kittiwakes. Murres are large seabirds, wing-propelled divers, able to commute considerable distances from breeding colonies and still successfully incubate eggs and raise chicks. This is a thick-billed murre and a common murre, very closely related species, and both are very capable of diving all the way to the bottom. They're really thick-bodied birds, pretty much built like bricks. They weigh about 2 pounds, so just about the same as these 2 pounds of butter. You might be more familiar with some of their smaller relatives, puffins, tufted and horned puffins, and these are also nesting at the cliffs, but our focus was on the murres and then the kittiwakes, which is a medium-sized gull, about the same size as our mew gulls here in the interior, but with their wingtips tipped in black. The Cape Thompson seabird cliffs are just phenomenal, just really spectacular. And we stayed at Ogaturk Creek, just south of the seabird cliffs, and this is what was called the Project Chariot site. For those of you who aren't familiar with Project Chariot, it was a proposed nuclear blast that was actually 20 times stronger than either of the bombs that the US dropped on Japan in World War II. Why? Edward Teller and the Atomic Energy Commission thought it would be interesting to try to build a deep water harbor, just a random experiment. Well, eventually the site was abandoned without any cleanup. Cleanup actually occurred quite a few years later. There's a great suite of environmental studies that was done from 1959 to 61, including a study on seabirds by Jerry Swartz here at UAF. Project Chariot fortunately was canceled, and why, as you might expect, people started to think more and more about the dangers of radioactive fallout in nearby villages. For example, Kivalina to the south and Point Hope to the north, and also beyond. I think there would have been just a devastating impact on the seabird cliffs, including landslides and tsunamis. These cliffs are limestone and crumbled enough on their own. There is also political fallout. Les Viereck and Bill Pruitt were two young UAF professors doing research out there, and they were fired from the university for voicing their concerns. If you're looking for a great read this summer, pick up Dan O'Neill's The Firecracker Boys, which tells us about this story. The summer of 1978 was the first that I went to Cape Lisburne, the very northwest tip of Alaska, 40 miles north of Point Hope. There's an Air Force station at the east end, so that provides great access to and from the cliffs. There are about 200,000 murres and 15,000 kittiwakes nesting at this colony. And if you look at the figure on the lower left, it shows ocean currents through the Bering and then Chukchi Sea, and you can see that every arrow goes north. And actually these currents come very close to the coast right by Cape Lisburne. So these currents are basically conveyor belts of food passing by, and they're passing very close by Cape Lisburne. And the colony, in a very good breeding year, especially once there are chicks to be fed, really stinks, and it just reeks of productivity. Another thing to note here is that from Cape Lisburne to the Canadian border along the Chukchi and Beaufort Sea coast, there are absolutely no cliffs. So this is, even though there's lots of food, farther north in the Chukchi and in the Beaufort, there's simply no place for these cliff nesting birds to breed. So the general outline of the talk is that I'll first talk about some key and different features of birds, and then focus on two species who stay the entire winter, and then look at various aspects of migration. This just shows some photos of our Alaskan birds, a golden eagle, a ruffed grouse, and a Pomeranian Jaeger. Birds and mammals have independent origins. Mammals evolved from therapsid reptiles long ago, about 200 million years ago. So if you traced back far enough in Ancestry.com, you would find a beast kind of like that in the upper corner as one of your ancestors. Birds evolved much later, and they evolved from dinosaurs about 150 million years ago. And this just shows the closest relatives of birds are the dromaeosaurids, or the velociraptors and their relatives of Jurassic Park fame. A more distant relative is Tyrannosaurus rex. So just some key differences between birds and mammals. One is that birds are adapted to powerful flight, especially when we compare them to bats. When we look at a bird hand and fingers, the bones are fused and reduced, and they provide the attachment for the 10 or so primary flight feathers, the outer flight feathers. The flight muscles, as you know if you looked at a chicken wing versus a chicken breast, are concentrated in the core of the body, and they're large and attached to a keeled breastbone. What I've shown here is a skeleton and a diving puffin, and so they're wing-propelled divers with large keels, massive breast muscles. This enables them to be strong flyers in both air and water. In the news, there was recently an article about a field study of color vision in broad-tailed hummingbirds. And it was documented that these birds have 4 cone types. Well, actually, all day active birds have 4 cone types. With non-overlapping peaks in sensitivity, they see all the wavelengths we do, but then they also see into the ultraviolet wavelengths that aren't detected by our eyes. Species that are nocturnal, like owls, have actually lost all or part of or all of that color vision. They have very few cones, and they have very densely packed rods. And why do they have this? Well, rods are 100 more times sensitive to light, which is very important, of course, if you're operating in near darkness. Actually, the 4 cone types is the ancestral condition. Reptiles also have 4 cone types. And it's thought that the reason that mammals, including people, only have 3 is that early in our ancestry, primates were generally crepuscular, operating at dawn and dusk, or nocturnal. So we've lost some cone types relative to our distant ancestors. And so then what we see versus what birds see the world painted very differently is one way we can think about it, and emphasizing features important to them. If we look at some renditions of what people think bird vision is compared to human vision, it almost seems like birds are having a psychedelic experience compared to us. Birds have a high body temperature. Typical mammal is like us, 98.6 degrees Fahrenheit. A typical bird is actually about 9 degrees Fahrenheit higher at 106. This is near lethal. A little higher body temperature and proteins start to degrade, whether you're a bird or a mammal. But also, another aspect of this is that this isn't trivial in how fast their engines run compared to ours. At rest, birds are metabolizing about 30 to 40% faster than we are. So how can they maintain this high level of metabolism, even higher than ours? Well, one big advantage is that, like us, they have four-chambered hearts, but in comparison to mammals, they have a large heart, on average, 41% larger than a mammal of the same body size. The largest relative to body size, as you might expect, are the hummingbirds. What I've shown here is a photo of a rufous hummingbird, which is the most northern and the one that you're likely to see if you get down to south coastal Alaska. Heart rate of birds compared to mammals is relatively low at rest, but high when active. And again, hummingbirds take this to an extreme. Their hearts are beating at more than 1,200 beats per minute when they're flying. Mammalian respiration, if we look back to our ancestral reptiles, they have alveoli in the lungs, but they don't have a diaphragm like we do. The diaphragm is a real key to improved gas exchange compared to our ancestral reptiles. The efficiency, though, is only about 25%. And so what I mean by that is that for every 100 molecules of oxygen you breathe in a breath, you're only taking about 25 of those out. The rest are just too far from the bloodstream in these dead air sacs, these alveoli, for you to pick it up. Bird respiration is just completely different. They don't have alveoli, they don't have a diaphragm, so what's going on? Well, they have air sacs, a large series of air sacs, but there's no gas exchange in those air sacs. Those air sacs are like balloons that deflate and inflate with no gas exchange. Then in the lungs themselves, they have what are called parabronchi, the PBs in this figure in the corner. These are porous tubes and literally just soak up oxygen as it's breathed in. The real key feature is that there's a unique one-way flow of air through the lungs. So what happens if you're a bird and you breathe in, that air isn't going to your lungs. That air is going to your posterior air sacs. Then through the lungs, then through the anterior air sacs, and then finally expired. It's actually a two-breath cycle. When birds are inhaling and inflating those posterior air sacs, at the same time, the previous breath is moving from the lungs and inflating the anterior air sacs. And this one-way airflow through the lungs greatly increases oxygen uptake, 50%. So twice as much as birds are extracting twice as much oxygen from each breath as we are. This greatly increases aerobic capacity. And the respiration, this two-breath cycle, it's really a bellows effect. The air sacs are simultaneously inflating and then deflating, the posterior and anterior air sacs. And that's synchronized with the wing beat when the bird is actually flying. So this is just so strange and so different. It's like, well, how could it evolve? Well, recent work on theropod dinosaur respiration based on fossils, as I've been saying, shows that theropods also likely had a one-way airflow through the lungs. Were they similar to birds in body temperature? Well, no. It's thought now that they were homeothermic, that they maintained a high internal body temperature, but that it was only about 90 degrees Fahrenheit. But with that one-way airflow through the lungs, those velociraptors must have had great stamina. Is it an insult to be called bird-brained? Actually, the brain of birds is large and well-developed. And there was a recent study of corvids, ravens and their relatives, and parrots versus primates. There are 3 comparisons in this figure here, and each of these shows that the bird brain is smaller, but actually has higher total numbers of neurons than the mammal that it's being compared to. So for example, in the middle, we see that a raven has a smaller brain than a capuchin monkey, but if you look at the total number of neurons in the brain, ravens actually have more neurons. They're just much more tightly packed together. And what this means is that birds are highly intelligent, and they're highly intelligent, high-performance animals. And one example that you will look at on your own, I hope, after this talk is over, I'd like you to Google goshawk flight video. Here's a goshawk. It's a resident of our forests. It's an accipiter, or a bird predator, and it's the largest of the accipiters. And has very prey, but it makes very high-speed maneuvers through the woods around Fairbanks. And if you look at this video, you'll just see how adept they are at making these, thinking and reacting very quickly. So the goshawk is one of about 30 resident species in the interior. The general problem, of course, is cold, dark winters. And these birds show quite a variety of both physiological and behavioral adaptations. What I've shown here are 3 of our residents, downy versus hairy woodpecker on the top, downy being much smaller with a smaller bill, and a Bohemian waxwing down below. I'll first talk about black-capped chickadees, one of the best studied songbirds in North America. They're small, weighing the equivalent of about 2 US quarters, and they're resident wherever throughout their geographic range. They're resident. They're not migrating at all. And they're in deciduous and mixed woods for the most part. And as I said, they're well studied. Susan Chawbaugh here in Fairbanks did her PhD studies on black-capped chickadee physiology at UAF. So one question, do chickadees fatten in the winter? This bird down below here certainly looks plump, but is it just fluffing its feathers, or is it fat underneath? Actually, the percent body fat of chickadees in winter is low, but with a distinct daily cycle. So in the morning, after they come out of their roosts, they roost individually through the night, whether it's, no matter what the temperature is. In the morning, when they start their activity, their body fat is about 2 to 3%. And then through the day, their body fat builds up as they forage. But it only gets up to a maximum peak of about 7 to 8% in midwinter. And in fall and spring, on days that aren't so cold, they don't build up nearly as much body fat. So 7 to 8% body fat isn't very much, and it's not enough that they could take a vacation day if it's really cold. In fact, it's on the coldest winter days that they have to work the hardest to find enough food. The graph at the bottom shows air temperature down to -40, and on the vertical it shows feeding intensity, and just shows that the colder the temperature, the harder the chickadees are working. Torpor, you might think that it would be an advantage for the birds to go torpid. Torpor is a controlled lowering of the body temperature, typically overnight, and it's generally rare in birds. And in fact, where we find it most common, that graph in the upper corner, is hummingbirds, swifts, and nightjars. But none of these are remotely adapted to our winters. And one of these, for example, the frog mouse, you can hardly see that there's a bird next to that graph. They're pretty much the sloths of the avian world. And I'd also just like to mention that there's only one bird on the planet that's a seasonal hibernator, and that's the common poorwill in the South US deserts. This shows a poorwill, and it's the purple in the graph is its winter distribution. So I said it's rare in songbirds, and in the songbirds in which it occurs, they don't seem to be dialing down their thermostat nearly as much as some of these other birds like the poor will. The body temperature only gets down to about as low as about 75 degrees Fahrenheit. Well, how about the black-capped chickadee? They do have that overnight torpor or overnight hibernation. And when they do, on the coldest nights, they're reducing at about 20 degrees Fahrenheit to a body temperature of about 89 degrees Fahrenheit. That may not seem like a huge drop to us with our normal body temperature of 98, but remember that they're going from about 106 degrees Fahrenheit down to the 89. This actually saves them quite a bit of energy, about half the energy they would have spent if they had maintained that high body temperature through the night. Recent work with the boreal chickadee indicates that they've reduced their body temperature as well and actually turned down the thermostat a little bit more to a body temperature of about 85 degrees Fahrenheit on the coldest nights. There's been some anecdotal observations that boreal chickadees survive the coldest winter nights, minus 50, minus 60, more than black-capped chickadees. And this may be one of the factors that contributes to their slightly better survival in the coldest conditions. Chickadees cache foods, as all of you know who have bird feeders, and they're stashing individual items separately over their home range, and for example, in crevices and tree bark. We know from laboratory experiments that their memory to recover those caches is excellent, at least overnight and possibly for much longer. There have been some studies comparing black-capped chickadees from different localities, and here's one that compared birds from Anchorage versus Fort Collins. And so the setup was they put these birds in captivity and had this experimental setup that's shown in the upper corner. There are 70 potential caching sites, covered tubes, and the birds were allowed into the room to either feed or cache, and then after they had been in there for some time, they were removed and then put back in the room either 5 or 24 hours later to see how accurately they could recover the caches they had made. If we look at these two graphs, the Anchorage is green, Fort Collins is red. The top figure shows eating and caching rates. So this shows that the Anchorage birds cached over twice as many seeds as the Fort Collins birds. And then the lower graph shows cache retrieval accuracy. And this is the vertical here is actually the number of looks it takes to recover a cache. So the fewer looks, the more efficient they are. And the Anchorage birds, you can see, were much more efficient at retrieving their caches than the Fort Collins birds. The hippocampus is the portion of the brain that's responsible for spatial orientation and memory. In seed caching chickadees, the hippocampus actually enlarges, and enlarges quite a bit with caching experience. This graph in the lower left here shows 3 different species that are closely related to the black-capped chickadee, including the mountain chickadee, which is shown up above there. And birds, this labels are pre-experiment, inexperienced, and experienced. So the pre-experiment are adults who have not done any caching. The inexperienced are young birds that have not done any caching. And you can see that their brain size is just about exactly the same. But as soon as you allow a bird to start caching and retrieving, the hippocampus starts to increase in volume, and it goes up, as I said, about 30%. There is a study of geographic variation in the size of the hippocampus of black-capped chickadees. And they took birds from Manhattan, Kansas, all the way up to Fairbanks. And they also had various measures to rank winter severity. And of course, Fairbanks came out right on top with the harshest winters. Well, what's the volume of the hippocampus of our birds compared to other birds? Well, I've circled the Fairbanks birds in the 2 graphs here. The Fairbanks birds have the highest volume of hippocampus, so their hippocampus increases more in volume than birds who are farther south. And then they also have the highest number of neurons in the hippocampus. So you might think that our chickadees are the smartest. What I also think is that it just shows that there's a real premium on caching in a harsher winter environment. The other resident I'd like to just briefly talk about is the American dipper. The scientific name is Cinclus mexicanus, which is really a misnomer for our birds up here. Their distribution ranges from the northern Brix Range well into Central America. In Alaska, their habitat is ice-free sections of fast-flowing streams. And I want to emphasize that they definitely are resident. They don't move at all. Pairs defend territories year-round wherever they're found. They're walking and eating invertebrates on the bottom, both summer and winter. And in winter, they're hyperactive. They're typically diving 5 to 10 times a minute when they're feeding, and they are well insulated. Somebody went to the trouble of counting feathers. And on average, they have about 4,200 feathers, and for a bird that size, you'd actually expect about 3,000. And a lot of these are down, so it definitely makes them better insulated. But still, they're going to have to shiver or work to keep warm and keep a high constant body temperature in cold winter temperatures. One neat adaptation is their eyelids are feathered, and that's the white that shows in this bird in the middle. Overnight, they use sheltered roosts. And as far as we know, even though the pairs stay together for years, if not for life, they seem to roost individually. And you'd think, well, it'd be a great advantage thermally if they could roost together and keep each other warm a bit. It's not known if our American dippers do this at all, but a related species, the white-throated dipper in Europe, it's been documented that they sometimes roost together in pairs and sometimes as family groups. So the dipper habitat in Alaska are unfrozen stream stretches that occur regularly and predictably year-round, year after year, because remember, they're spending their entire lifetimes there and they have to be able to feed every day. Here's an example of Schist Creek in the Alaska Range where Ken Whitten and I skied in one winter and found dippers in these types of habitats. And the American dipper, it's my vote for the greatest ability to thrive in the harshest winter conditions, because they're either feeding in near freezing water or they're out of the water in much colder air temperatures. Let's switch gears here and talk about black guillemots. This is a somewhat smaller relative of murres and puffins. Normally, these nests in crevices in talus slopes, but on Cooper Island, a barrier island near Barrow, Utqiagvik, George DeVoke was doing breeding bird surveys in the late 60s and early 70s, and in 1972 found them breeding in what we would loosely call nest boxes. There was lots of debris on the island left by the Navy several years before, and the birds found these and found that they were suitable places to nest. So George worked here on his PhD at that time, and we talked about adding nest boxes to see if he might attract more birds. Over the next few years, he put out more and more nest boxes until there was a total of about 200. He found that they were quickly occupied by black guillemots coming from who knows where. The closest colonies of any numbers are hundreds and thousands of miles away. And in the early years, non-breeding adults were abundant at the colony. And we talked about it, and it seemed like he could have put out 500 nest boxes and they'd all be occupied, but that just would have been too many to monitor. Because one thing that he's done is to band every fledgling at the colony year after year, and he's done this every year since. In fact, today he's heading out to the colony to start the work this year. And most of the adults he had individually color banded as well. These two individuals that I picked up are two birds that died in Cooper Island, and you might not see it very well, but each of these has a U.S. Fish and Wildlife Service band and a series of color bands for individual recognition. And I'd just like to say, it's very rare that you can pick up an individual, look at it, and know something about its individual life. So one of these was a male that was an immigrant that George banded in 1987. In 1988, it bred at nest N5 with a female who had been breeding at that site since 1983, and her mate didn't come back in 1988, so this male then mated with her. This male was found dead in 1989 and was either a peregrine or an owl kill. The other one also was an immigrant, and it was banded as a floater in 1983. So a floater means that it was an adult, but it didn't have a breeding site. In 1984, it started using an area with a large collection of rusting oil drums, and these were just then rusting enough to have holes in them that some birds decided were perfect as nest boxes. And it first bred in a rusty oil drum in 1986 and bred there again the next 2 years, and then died again in 1989. Having these color-marked birds, individually marked birds, just greatly increases our ability to understand what's happening in these populations. George's early work was focused on breeding success, and there are 3 essential ingredients. One is some type of a nest cavity. You can see they don't seem to be real choosy if they're using oil drums, but they need some type of a cavity. And typically they have a 2-egg clutch. And you can see a 2-egg clutch here, and they don't bring any nesting material in, but they do seem to like to arrange pebbles in a fairly artistic fashion. The second thing they need is at least 80 snow-free days to complete the reproductive attempt, because they won't dig through snow to get back into the nest site. And at Utqiagvik, Barrow, and Cooper Island, it was only in 1972 that summers had more than 80 snow-free days. But after 1972, just about every summer has had more than 80 snow-free days. And the third key ingredient is Arctic cod, a key prey nearby. George just compares kind of early versus later phases of this colony development. And it really went from boom to bust, representing global change in a heartbeat. From 1979 to 84, on the left here, the sea ice was near the shore in late summer. And what this figure shows is light blue is 15 to 30% sea ice, dark blue is greater than 30% sea ice, and white is actually open water. So you can see in 1979 to 84, there was sea ice right near the colony, even in late August to early September. And what this meant is that Arctic cod were very abundant near the colony, because they're most abundant in about 50% sea ice. Basically, everybody raised 2 fledglings every year. It was just a great time to be breeding there. If we contrast to that to more recent years, the part on the right here shows 2003 to 2012. In those years, you can see that there's no blue anywhere close to the colony. These are years when the sea ice, by late August, early September, was at the continental shelf edge, far from the coast, or even farther from it. Black guillemots are not very good about commuting long distances and coming back to the colony with food. They attempt to find food very close to the colony, unlike the larger murres, which will go a considerable distance. And with the sea ice far offshore, the adults couldn't bring any Arctic cod back, because they wouldn't travel to areas where they would be. And they were bringing sculpins back to their chicks. Sculpins are like nasty for the chicks to swallow, especially compared to the Arctic cod. And in these years, there's been very low reproductive success. And this has continued since 2012, pretty much every year has been like this. Geolocators are a relatively new technique of tracking movements of birds and other animals. And what geolocators are doing is recording light level, for example, every 60 seconds. And this provides information on latitude, just duration of the light versus dark period every 24 hours. And they also provide information on longitude if there are shifts in the light versus dark periods in that 24 hours. So for example, if you went from here to New York, there would be a shift in the light period you're experiencing. The drawbacks compared to satellite transmitters is that they provide relatively imprecise estimates of location. And a big drawback is that you have to retrieve them to obtain the data. You have to recapture the bird and remove the equipment. And a lot of times it's hard enough to capture a bird in the first place, let alone recapture it a second time. But the advantage is that they can be built much lighter, to about 0.5 gram, so that they can be put on even a bird as small as this warbler above here. The lightest satellite transmitters, by contrast, even now weigh about 10 grams, which is about the body weight of the upper bird there. George put geolocators on these black guillemots. These were programmed to record light level every 60 seconds. And the nice thing about guillemots is that you can capture them, recapture them, and recapture them, and they don't seem to be bothered too much. And what he found is that this looks through the winter from late October to late April, and they first go north. And they're going north into that about 50% pack ice. And then as ice forms in the Beaufort and Chukchi, you can see that the distribution gradually drifts south, but they're staying in that about 50% ice the whole time. And this is basically true the entire winter, that as ice forms farther and farther south, they're drifting farther south, but they're never getting out of that broken ice zone. This shows it quantitatively. With the green is the amount of ice 1 month pre-occupancy, 1 month before they were using the area, for example, the Beaufort Sea here. And then blue shows the ice concentration when they were actually at that site, in that location, and the red 1 month later. And of course, in the fall, the ice is expanding in this entire region. But what you can see is that it doesn't matter what month of the year, throughout the entire winter, they're finding about 43% ice habitat. And the reason for this is that's where Arctic cod are abundant year-round. So they have a ready food source as long as they can maintain themselves in that about 50% ice habitat. I'd like to go to contrast that, the Arctic tern, which many of you, if you were on Jeopardy and were asked, well, what bird makes the longest migrations in a year, you would say, oh, Arctic tern, and be rewarded for that. In Alaska, they're breeding throughout the state, but they're most abundant in coastal regions. These are really buoyant flyers. These squiggly lines at the bottom are showing the tracks of birds that have been outfitted with geolocators in the North Atlantic. And they're making an annual round trip between the Arctic and Antarctic and back, but pretty much in a leisurely way compared to some of the other birds I'll talk about next, because they can feed as they go. And they especially will slow down that rate of movement to Antarctica if they find a hot spot where feeding is especially good. From a lifetime perspective, they're traveling unbelievable distances. And here's a quote: "As Arctic terns can live over 30 years, the total distance flown in a tern's lifetime may exceed 2.4 million kilometers. That's 1.5 million miles. An equivalent would be 3 round-trip journeys to the moon." The bar-tailed godwit is a medium-sized shorebird with an upturned bill. They're large enough that they can be fitted with satellite transmitters. And this figure shows in the middle this bird A9. you see that, you can actually see the antenna coming back from her tail. So what are these guys doing when they're migrating? Well, the Alaska subspecies boweri that breeds in western and northern Alaska, they winter in New Zealand. And then in the spring, they migrate north, become via the Yellow Sea. And these are the guys that are the world record holders for nonstop flight, continuous flight. In the fall, they fly day and night, no rest, not landing on the water, from the Alaska Peninsula to New Zealand. Takes them on average about 6 days to fly about 7,700 miles. One thing they do before leaving is they about double their body weight, getting up to about 41% body fat. Might think it's hard to get off the ground in that condition. When they do leave, they exploit favorable tailwinds, and this certainly helps them make this long journey. Here's the annual cycle of one bird with a satellite transmitter, E7, a female. She made 3 nonstop flights in the year. First in the spring, she traveled 6,300 miles, taking 7 days, to the Yellow Sea. And then she made a 1-month stopover in the Yellow Sea, recovering and rebuilding her reserves for the next flight, which was a 4,500-mile flight in 6 days back to the breeding grounds in Alaska. Then in the fall after breeding, she made the 3rd flight, the nonstop flight back to New Zealand. She went 7,200 miles in 8 days. So she traveled in that year about 18,000 miles, and all of it nonstop flight. In the news recently, Ned Roselle in his Alaska Science Forum wrote an article about blackpoll warblers. He was with his wife, Kristen Bartecki Roselle, as she was doing a breeding bird survey near the Tanana River in South Fairbanks. And they heard and saw a singing male blackpoll warbler, and they took that picture that's in the middle there. The title of his piece is World Traveler Visits South Fairbanks. Kristen's master's focus when she worked here with me at the university was on yellow-rumped warblers, a very closely related species and just about exactly the same size. So I don't have a specimen of a blackpoll warbler, but this is the size of a blackpoll warbler. Again, like black-capped chickadees, they weigh about 12 grams. Their breeding habitat is in black spruce forests throughout their range. Beyond tree line, like out on the Seward Peninsula beyond tree line, they also nest in tall streamside thickets. It's an example of what we call a neotropical migrant. So a neotropical migrant is a bird that breeds in North America and then overwinters in either Central or South America. And these guys are actually overwintering in northern South America, so they're a true neotropical migrant. And they're what we call a nocturnal migrant. They're migrating at night. And this is very typical for these types of birds. If these birds aren't migrating, they're sleeping through the night pretty much like you and I would. They go through cycles of REM and slow wave sleep, brain waves very similar to what you see in us when we're sleeping. The apparent migration route of the blackpoll warbler is that in the fall they go almost due east to the northeast US, to the Atlantic coast, and then over the Atlantic Ocean to South America. And then in the spring, it's thought that they take a much more direct route back to breeding grounds. There's a conservation concern for this, well, and many other species too, but it's one of the species whose numbers seem to be declining the fastest in North America. This graph shows this line that you can see from 1970 to 2010 shows the index of numbers of this species, and you can see that it's gone down a lot just in a few decades. So it was felt that there was a need to verify their migration routes to help conserve the species, and the way to do this would be to monitor the movements of known individuals. They're way too small for a satellite transmitter, of course, but the geolocator technology has gotten small enough that these birds can be equipped with geolocators. Bill DeLuca published the study in 2019 based on his work of attaching half-gram geolocators on blackpoll warblers at 4 different sites ranging from Churchill to Nome, Churchill, Manitoba to Nome. They only put them on breeding males. So You might ask, well, why just breeding males? We'd like to know what females and juveniles are doing too. But first, adult annual survival is about 50%, higher than that of first-year birds, which is generally in the range of about 20 to 30%. So a much better chance of having an adult come back the next year. And also, males are much more likely to return to the same territory or very close territory 1 year after the next. And then the real key is that these are the ones that can be captured and then recaptured. Remember, you have to recapture the bird to get the geolocator back off to obtain the data. And the adult males on breeding territories can be captured and also recaptured using playback of male song, because they'll act aggressively, and mist nets. You don't want to do this normally because the playback of song really does disturb the birds, but this was the only way that they could both trap and retrap them to do the geolocator study. One thing I'd like to say is that the geolocator weight is about 5% of their body weight. So to put that in perspective, that would be about the equivalent of strapping this 10-pound bag of sugar on my back. Without it, of course, I can still dunk a basketball. But you stick this 10-pound bag of sugar on me, and there's no way I can do that. So they managed to get 111 geolocators on these birds at these 4 different locations, and they were able to get 27 of them back. That doesn't seem very good, but remember, male survival is only about 50%, so you'd only expect about 50 to 60 even in the best of situations. But 6 of these failed before migration, 7 provided data on part or all of the fall migration only, and only 14 provided data for the complete fall and spring migrations. But those 14 really do provide a lot of information for us. So fall migration, step 1, it is to the Atlantic coast, as everybody thought. On average, it took these birds 18 days, ranging from 7 to 29 days. The authors didn't break this down by location, but we might expect that the birds from Nome would take the longest. So just to think about how much they have to travel each night, the distance from Nome to Boston is 3,800 miles. So even if they take the longest 29 days, they'd have to average about 130 miles a day. Flight speed of these birds is about 30 miles per hour, which would mean these birds traveling from Nome, if it's going to take them as long as 29 days, they still have to migrate for more than 4 hours every night on average. But why go east instead of more directly south? They come more directly south when they're coming back in the spring. And I think it helps to have a historical perspective on why this might be. If we look at spruces from 21,000 years ago until now, if we start with the 21,000 years ago in the far left there, spruces were distributed in eastern North America, south of the continental ice sheets. And then gradually started spreading to the west on the south side of the continental ice sheets. And then as the ice sheets started to melt away and you get that opening between the two, spruce gradually, the distribution of spruces started heading northwest, and then finally spruces were abundant in Alaska by about 9,000 years ago. So I think that the changes in the blackpoll warbler distribution likely coincided with those of spruce because they're so wedded to spruce. And that Nome was likely colonized last. And so I think what we're looking at is that the westernmost birds have just simply extended an ancestral route, and then they're still following that. How do these birds navigate? Young birds are migrating independently of adults. So how do they find their way? This hasn't been studied at all in blackpoll warblers or related species, but I think there are some possible analogs. One being the garden warbler in Europe. It's what we call an old world warbler, a different family than the blackpoll warblers and other wood warblers, but they do migrate at night and the young migrate independently of the adults. Eberhard Gwinner did studies of these and other old world warblers, and he took advantage of what we call Zugunra, or migratory restlessness of caged birds. If you have birds that are migrating at night, and it's not the time of year to migrate, these caged birds will show no restlessness, and they'll go through a normal full night sleep cycle. But if local birds of the same species are migrating, what you see in these cages is that these birds attempt to fly in exactly the same direction at the same time of night and for the same duration of birds in the wild. It's like they're programmed to do exactly the same thing we're seeing in the wild. Some of Gwinner's birds he captured in Germany. The garden warbler migration from Germany in the fall is to first go southwest through Europe, and then at the Straits of Gibraltar make an abrupt 90-degree turn and fly into this area of Africa where they're going to winter. And then in the spring, they follow a different route. They go directly north. What happens with these young birds in captivity? Well, they show, this is on the bottom graph here, they show migratory restlessness on the same nights the wild birds are migrating and in the same direction. And they make an abrupt change about the 1st of October from going southwest to southeast. And then in the spring, their orientation is roughly due north. Garden warblers certainly have a genetic program on both when to start and what directions to migrate. Do blackpoll warblers have a similar program? Well, as I said, we don't know. Nobody's studied it the same way. But if you look at these graphs of, this is migration southward, which is really eastward, as we know now. The first departures from Nome and Denali were in mid-August. And in the upper figure there, that's the arrow. That's earlier than anybody had documented the start of migration within the species. So these birds are in the far west that have the farthest to go are leaving earlier. But like all the others, they're arriving in South America at about exactly the same time. So this is an example of directional natural selection. So there's variation in departure dates in any population, and you can see that even with the small samples, the birds from Nome, Denali, Whitehorse, and Churchill. Not everybody is leaving at exactly the same time. And in the West, there's likely a premium for leaving earlier in mid-August than it would be for birds in Churchill. And I think the birds that are leaving in mid-August are going to be those that are more likely to survive the winter and return successfully. And all this means is that just a gradual tweak in the genetic program on when to start migration as the species expanded west and north after the last ice age. The second step of their migration is not a migration. I might say this is a vacation on the eastern coast of North America. On average, they're staying there 26 days, but quite a range from 18 to 41. And like those godwits, they are exploiting favorable tailwinds, but they have to leave the northeast US or the eastern coast with all the fuel they need for that entire trip. So flight distances are up to 2,200 miles. In this particular study with these birds with the geolocators, they found that their flight distances ranged anywhere from 1,400 miles if they were coming from farther south along the Atlantic coast to 2,100 miles if they were coming from farther north. The birds from Nome are the orange bars in the figure, and they're the birds that traveled the farthest and took the longest because they're the birds that were coming from farthest north. Their spring migration is a totally different route and much quicker overall, averaging 34 days. And if the birds have longer to travel, they start earlier and all arrive at their breeding sites about the 1st of June. So you can see from the graphs on the left, it shows the start of migration. The birds that will be going to Nome are starting in mid-April, as is only one left from Denali that was carrying a geolocator. Birds from Whitehorse starting almost the same time, but birds from Churchill starting later. And I think what this represents again is directional selection. to leave earlier if you have a longer distance to travel to get back to the breeding area. The Pectoral Sandpiper is a medium-sized shorebird, and the last example I'll talk about. They have varied migration routes and distances. They're wintering in South America and then coming to the high Arctic to breed. And they might see them passing through Fairbanks on spring and fall migrations. This is a species that's polygynous. One male may mate with multiple females. And it's also a species that's what we call a lek breeder, like our sharp-tailed grouse here in the interior. What I mean by that is that males defend small display territories very close to each other. And then females come to that lek and pick one of those males as a mate. The males don't incubate at all. Only the females incubate in this species. In many other shorebirds both incubate, but some they don't. And this is one where only the females incubate. Bart Kampenaers, working near Utqiagvik, looked at numbers of males and duration of male stays there compared to the numbers of females. And so the more females that were there in a particular year, the more males were there too, which you might expect. But then he also found he had individually marked males, and he found the ones that stayed longer fertilized more eggs. And so that begs the question, why doesn't everybody stay longer if staying longer helps you sire more young? Well, when Bart was just wandering around one evening, he observed male pectoral sandpipers departing Point Barrow and flying north over the broken pack ice where there would be no food, and he wasn't finding any returning. So the question would be, well, what are they doing? So again, he put geolocators on some and found a real surprise. The males that were displaying at Utqiagvik, some of them later moved up as far as 8,100 miles to other breeding sites in the north in the next 4 weeks. Some of them went up to as many as 23 additional breeding sites during that period, again, looking for potential mates. And this figure just shows just how far these birds are traveling. And normally when you see a bird breeding, you think, okay, it will breed here, then recover, and then migrate south. But these birds that are breeding around Utqiagvik, that's just the start of their breeding season. After that, they may be traveling incredible distances while still attempting to breed. And that's all for today. We'll certainly face more and more challenges to conserve birds in the near future, and hopefully our advances in our ability to track birds will continue and aid in the effort. I'd like to thank the Department of Biology and Wildlife here for the bird specimens, and also the Alaska Songbird Institute, which is doing much of the migration research here in Fairbanks. Thanks.
Thank you, Ed, for increasing our understanding of our birds this evening. And thank you to KUAC for hosting the Discover Alaska series. If you would like to see this lecture again or to let others know about it, you can find it on our website at uaf.edu/summer/events. Join us next week to learn more about the U.S. Census in Alaska. I'm Althea St. Martin, and thank you for joining us as we discover Alaska. Have a wonderful evening and stay safe.